Okay, I'm picking up the rear end today. My name is Michele Nishiguchi, and today I'm going to talk about ecological diversification of Vibrio fischeri during the planktonic phase and subsequent consequences for squid host colonization. And I just want to actually acknowledge my two collaborators here, Will Soto, who was a previous PhD student of mine. He's now working with Mike Travisano as a postdoc, and Mike Travisano. Okay, so ecological diversification is very interesting. People have been talking about all morning long in terms of looking at how bacteria are a really nice model system to look at both biotic and abiotic factors that drive speciation. And a lot of times you can test these things in the lab quite easily just based on the fact that bacteria has really quick generation times. And it's really nice because you can look at how it initiates adaptive radiations. So here I just have two figures from Chris Mark's paper and another one by Myers and Berbring, just showing that depending on the number of ecological niches presented to an organism can drive specialization in many different ways, and you can get clonal interference where you have upon generation after generation, lots of variation in the kinds of phenotypes and possibly genotypes that you see. Work was initially done by Mike and Paul Rainey way back 16 years ago, looking at bacterial adaptation radiation in Pseudomonas fluorescens, and they found that when you just, upon growing a single clone, that eventually adapts into 3 different phenotypes, and this was a smooth morph originally shown here, and then a wrinkly spreader, and then a fuzzy spreader. So I've been working on a model system using Vibrio fischeri and bobtail squid as a model system, and people actually really haven't looked at adaptive radiation of Vibrio fischeri very much, and so we've decided to kind of take that track. And we've noticed along the ways of looking at many of our strains that there's a lot of differences in colony morphology, and possibly that could affect host colonization. And one of the things that Will discovered, I called this the forget your culture test tubes on the bench for 2 weeks experiment, but he noticed that you can eventually develop these wrinkly spreaders on a plate. And we wanted to test whether these were better or worse at colonizing host squid. And they're actually derived from heterogeneous microcosms, so you just start off with a smooth morph, and eventually you get a lot of variation in these wrinkly spreaders over time. So let me just tell you a little bit about the model system that we work on. So it's bacterium Vibrio fischeri, which is a gamma proteobacterium, and Euprymna in the squids in the genus Euprymna. It's a beneficial association, so actually the bacteria provide light for the squid, and I'll mention that in a little second. And the interesting thing is these bacteria are environmentally transmitted through every generation. So the squids, when they're hatched, they don't have these bacteria, and they acquire them anew from the surrounding seawater. So that allows a lot of leeway for what kind of symbionts they pick up, and the selective forces that are acting upon the bacteria, whether they're environmental, as they're floating around in seawater, or once they're inside the squids. And we can look at diversification of endemic strains because there are many different genera of these squids around with multiple strains and species of Vibrio fischeri and a few other Vibrio species along the way. So in this symbiosis, you can find bacterial symbionts, at least Vibrio symbionts, in 2 families of squid. The Sepiolidae, which are the bobtail squids, what I'll focus on today, but also a few of the species in the Lolaginidae, which are more likely what you'll find on your plate of calamari as you order it as appetizers tonight. So, but when you do a ventral dissection of the light organ in the mantle cavity or the body of the squid, most of the squids that have light organs are bilobed. So you can see there's 2 lobes here. This is the body cavity, the eyes and the arms would be way up here. And the black is the ink sac that surrounds this light organ. And the bacteria are housed in the specialized light organ. And upon closer inspection, this is a transmission electron micrograph. There are many different channels, or we call them crypts, that the bacteria are encased in. They're extracellular, so they're not inside any squid cells. And these little electron-dense particles here are single Vibrio fischeri bacteria. So the other thing that makes this system very tractable is that the squids, you can bring them into the lab live and raise the baby squids. So you can hatch them, and they're born exenic, so they don't have any of their symbionts. And you can present to them a variety of different wild-type bacteria or genetically modified bacteria that you want to test to see about competency. So this is the beauty of this system. So depending on the species that you work on, they have a 2 to 6 week developmental time. They're direct developers, so they don't have any larval stage. And they're born, like I said, aposymbiotic, and they acquire their B. fischeri bacteria within the first couple hours when they're hatched. And from then on, they become symbiotic, so they stay symbiotic throughout their life history, unless you present antibiotics to them. And then one of the interesting things that these squids do, every morning with the onset of sunrise or light, they vent out 95% of those bacteria. And the remaining 5% recolonize the light organs. So it's somewhat of a bottleneck effect because they're just swishing out most of the bacteria and the remaining 5 stay in there. And I should say those 95 that get vented out are also viable. So they're swimming around in seawater ready to colonize new generations of baby squid. So I said these bacteria produce light, they're bioluminescent. And so what the squids do is during the day they bury in the sand and hide from predators. And this is just a bacterial growth curve that's shown just to show you what the bacteria are doing at the same time. And then right at sunset, they're going through log phase during the day. They reach kind of a stationary phase where they're in form, so they can actually produce bioluminescence. And they can make light. So the squid unburies at night because they're nocturnal, and when they're hunting or feeding, they use this light production for a behavior called silhouette reduction. So if you can imagine in clear tropical waters, downwelling light coming down on a squid, it's going to make a shadow from below. And so what the squids do is they match the downwelling moonlight with the bioluminescence for the bacteria. So we call it the squid cloaking device, or stealth squid. Anyways, and so this is just a cartoon diagram of showing you what they do. And they do this, and then this is dawn. So like I said, they vent out 95% of the bacteria, boom, and then they bury in the sand, and the bacteria keep growing every day, every day. And this is just to show you that we've done these experiments in the lab. So here's actually environmental light. You can increase or decrease the light. You put your little squids over a luminometer, and they try to match the downwelling moonlight as you're increasing or decreasing the light. Okay, so when we start planting out these Vibrio bacteria, you notice a lot of different, this is from a single clone, a lot of variation in the morphology, so some smooth types, some wrinkly spreaders here. So, and Will noticed that when he actually got these wrinkly spreaders, when you just let your culture sit on the bench top for a couple of weeks, they start to form this nice pellicle. And so he was actually working with the bacteria that were actually making this nice biofilm form pellicle on top. And the smooth morphs, I should say, are swimming around here in their planktonic stage. And so here is just to show you a diagram what they look like. So here's a smooth morph that's still kind of in the planktonic phase, and then these wrinkly spreaders that are actually forming this pellicle. And this pellicle gets very strong. If you let that test tube sit on the bench for a very long time, you can actually flip it upside down. This is no tricks with the photograph, just to show you that we are really doing this. And that it can hold the media like that. I mean, it's pretty thick. I mean, it's really amazing. It's like, when I first saw this experiment, I was like, is that fungus in there? But it's actually all bacteria. And this is really interesting. What Will did then is he took a wrinkly spreader and then just started culture, and it forms this pellicle. There's no smooth morphs in here. They're just all pellicle on top. Okay, so to give you an idea, we're tying to the smooth morph over time. So this is 1 day, 2 days, and 3 days. It still stays the same in terms of what it looks like. And this is the wrinkly spreader, so the same time point, 1 day, 2 days, and 3 days is at, it's the same thing. It gets kind of more wrinklier. This pellicle actually really forms a stiff type of polysaccharide matrix. And then if you just do a cross-section again, same thing. This is a smooth morph, so there's not really much, there's pretty much homogeneity throughout the strain when you look at it from a cross-section. But the wrinkly spreader has this really complex amount of matrix and polysaccharide that's associated with that morphotype. So we took those two strains and we decided to do competitions because we have these animals in the lab, and we can say, well, what's the benefit of being a wrinkly spreader? They're making more polysaccharide in this biofilm. Does it give it an advantage in terms of being inside the squid and colonizing the squid? So we've done it with a bunch of our environmental strains, and so this is the first strain I'm going to show you is Vibrio fischeri from a fish host. Okay, so this is not a squid Vibrio fischeri, and this is the host Monocentris japonicus. It's the pinecone fish from Japan. And so the blue is just the smooth morph, and the red is the wrinkly spreader. And these are initial, we call them monocultures because these are single infections, but we put them on the same graph. So here's the smooth morph, and so the smooth morph can colonize. And this is not a squid symbiont. We're doing these colonizations in squids. So fish symbionts can get in, but they can't persist. That's just in general. We should do the wild type. So here's the wild type, and it gets in, but over 36 hours, it just can't stay in the squid. And the wrinkly spreader can get in, and get in at a little better, higher levels, but it still can't persist as well. But when you do the competition, so 50-50 presented to the squid at the same time, the smooth morph just gets knocked out, and the wrinkly spreader persists. But again, because they're not very good at staying in the light organ, they both kind of knock down by 36 hours. But this one is a much better colonizer when presented at the same time. When you do it with another pinecone fish, this one's from Australia. So I should say that these squids are, the Ecological tasmanicus from Australia. So now you're taking a fish that co-occurs with these squid, and they have Vibrio fischeri as well. So again, smooth versus wrinkly spreader. So these are the monocultures. So the smooth and the wrinkly spreader in this species of Vibrio fischeri from pinecone fish from Australia, they can actually persist. So they don't knock down like the Japanese Vibrio fischeri. And they act the same, and this is in monoculture when you do single infections, so just one in a squid or the other. But when you do the competition, again, the wrinkly spreader stays in, but then eventually they start dropping down. If you take it to 48 hours, they actually, again, can't persist because they're a fish symbiont. Okay, so let's go to the squids now. So we have squid symbionts now. This one's where ET is from the Australian squid, and ES is from our Hawaiian Euprymnoscolpes. So it's a congener of the host that we're testing it in. So these are the monocultures on the left, just to show you the squids. This is the Hawaiian one with Malay, and this is the Australian one with Shipwarney. So monocultures all act the same. So when you do single infections with either the smooth morphs from Hawaii or the smooth morphs from Australia, or the wrinkly spreaders from Australia and the wrinkly spreaders from Hawaii, they all act the same. And of course, since they're from squid, they can persist. Okay, so they actually persist. But when you do competition, and this is, now these are native strains. So this is native ET and Ecological tasmanicus smooth morph versus wrinkly spreader. Again, the wrinkly spreader does well. And these guys, this is going to be venting, and they're going to persist over time. So they'll get vented here and then keep going over the time course. And then when you do it with, again, this is the ES wrinkly spreader. So we know that ES is actually the non-native strain. Never wins in a competition. But these are competitions. So here's a native strain and the non-native strain. This is just smooth morph, so this is what you normally see. We've shown this many times. But then when you take the wrinkly spreader of the non-native strain and the smooth morph of the native strain, okay, it doesn't outcompete it, even though it's a wrinkly spreader because it's a non-native strain, but it does better than the smooth morph. So it's a little bit better competitor, but it still can't beat out the native smooth morph strain, all right? And you do the reverse when you do with the Australian wrinkly spreader versus the Hawaiian wrinkly spreader. It has the same results as it's just switched. And then finally, the last set is we took a free-living strain that can't colonize. So there's some deep fishery strains you isolate from seawater, say in off of California, where there's no squid, and you try to infect it so they can infect, but not very well. So this is actually an ATCC strain 7744. And so again, single infections. This is the smooth morph. This is the wrinkly spreader. They can get in, not the same levels, so colony form units. They just can't get in. They can't stay in. And but the wrinkly spreader, when you get an evolved wrinkly spreader, it does better. But again, it still can't stay in because it's a free-living strain. So again, wrinklies always win in a competition when they're at the same type. Just to follow up with bioluminescence, again, and this is just showing you wrinklies are in red and smooths are in blue, and just showing you from, this is Australian V. fischeri, Hawaiian V. fischeri. This is from the pinecone fish from Australia, and this is another strain from Japanese Euprymna, so Euprymna morsii. The wrinklies, again, have actually higher bioluminescence than the smooth morphs, and these are measured inside squids. So you can put your squid out and infect them into a luminometer to measure luminescence. And so the wrinklies actually make more luminescence, which is, if you think about what the squid wants more light to do the counter-illumination behavior. So you can see that, just taking a smooth morph, and you might get a lot of variation in terms of the wrinklies, and we've seen that just by, I'll show you the variety. We're coming up with lots of different variations. What is the genetics behind this? And so that's the next step. So hopefully the next talk that Will gives will be what we're going to find out. So we're trying to investigate those things, but it's really interesting to see this phenomenon. And I should mention that when you actually take a light organ from a squid in the wild, you don't see a lot of wrinklies originally. They're mostly smooth morphs. If you grind up a light organ from a squid from the wild, you just see smooth morphs. So why aren't these wrinklies more persistent inside the light organ? And we know they're related to biofilm, and that's very important for colonization. If you knock out biofilm genes, they can't colonize very well. So let me just summarize in terms of having V. fischeri in this planktonic phase and then eventually evolving to get the wrinkly spreader was really interesting because it affects the symbioses when you do colonization. And then the loss of motility, which is something I didn't talk about today. We do see a loss of motility in wrinkly spreaders, and that's actually been shown many times. When you increase biofilm production, you lose motility, and that makes sense because usually biofilm is associated with adherence, and motility is associated with trying to find where you're going to settle. And that's probably related to cyclic di-GMP, which we're investigating as well. Bioluminescence increased within the wrinkly spiders, and we know biofilm also induces quorum sensing, and that ecological diversification can override coevolution between hosts and symbionts. So what's happening in the environment, especially in environmentally driven symbioses, can also affect what happens later on inside the squids. So just in the future, we're going to look at different morphs. We're doing some selection experiments right now with lots of different variations of abiotic factors like UV, pH, and salinity. And then again, we're going to look at abiotic and biotic factors, how they affect adaptive radiation. And just a pitch for my student, Amy Nava, who's showing a poster today, because we've been looking at how grazing affects experimental wall biofilms as well. All right. And thank you.